We're at the 17th CROI. We're here with Dr. Koup, who is Deputy Director of the Vaccine Research Institute at the National Institutes of Health. And we're very appreciative of him to come from a meeting which, and it's appropriate that we do this because you did a more sophisticated presentation of monoclonal antibodies with the young investigators who are probably looking at all the fields that are out there and what's going on there. Maybe they made a decision, maybe they haven't, where they're going. But it's important for them to get a grasp of what this CROI is, and as well as what the fields are that they're in it. And we're many 300 steps down from there. We're basic, but we're interested. And the people that will tune into this program are interested in knowing what is monoclonal antibody. And maybe like we were just talking a moment ago, can you kind of start from the beginning of how you got there?
Sure. We're at the Vaccine Research Institute, so we're interested in vaccines. Antibodies are one of the major defenses against viruses. So these are proteins produced by the body to defend against any pathogen. They will bind to that pathogen and neutralize them, kill them, make them such that they can't infect. So as part of what we do to come up with a vaccine, we discovered these antibodies in individuals who had been infected for many years that could neutralize, wipe out many different strains of HIV. And so once we isolated these antibodies, we were initially intending just to use them to identify and characterize the targets on the virus that we would need to know in order to develop a vaccine. But what we ended up finding out was that these antibodies, because they were so broad and potent, they could actually be used for both treatment and to prevent infection.
Now, were these noticed in a lot of people or very selective people?
Well, it's a continuum, and it all depends upon how you define breadth and potency. But I would say that there are, on the high end, probably 20–25% of individuals after many years of infection make these types of antibodies, which are very potent against viruses. It's important to note that even though these individuals have those antibodies, their own viruses have escaped from those antibodies. So that those antibodies don't help them, but if we can isolate those antibodies and give them to other people, they may prove beneficial.
Now, why would that be? That doesn't seem reasonable, does it?
It doesn't seem reasonable, but it's what we've seen with HIV all along. And that is, HIV is able to mutate very rapidly. So whatever immune defense, once you're infected, whatever immune defense you throw at the virus, it escapes, whether that's killer T cells, antibodies, or anything else. And that's why HIV just continues to replicate in individuals.
So would there be resistance from these at some point down the line, or do you want to get enough of them differentially, slightly different to maybe collectively they make?
Yeah, I think it's very similar to the early stages of antiretroviral therapy when we had AZT and when we had D4T. We had just a couple of antiretrovirals. Any one of them led to resistance. The same thing is showing up with these broadly neutralizing antibodies. Any single antibody that's used, certainly for therapy, you tend to get escape. But if we can combine multiple antibodies with different specificities, we think we'll see the same thing that we do with combination antiretroviral therapy. We'll inhibit the ability for escape to occur.
So can you actually quantify the number that you might think you come out with? And was it going to be like 2 or 3 that you put together? Or like
I mean, we've done this in vitro studies and mathematical studies. It looks as though if you put 2 antibodies together, you'll cover 98% of viruses. If you put 3 together, you'll hit 100. If you put 4 together, you'll
more insurance.
You'll get even broader coverage. So yeah, I think 3 antibodies or maybe 4, and we should have everything covered.
Wow. So where are you in the stage of progress in this direction?
Yeah, so there are multiple different antibodies that have been made for use in phase 1 trials and have actually gone into phase 1 trials in humans. They've been shown to be safe. They've all been shown to have a half-life of about 2 weeks. And when given to individuals who are viremic, who have active viral replication, they will shut down viral replication by a log and a half for a week or two, and then the virus comes back up. And sometimes you start to develop resistance to the antibodies, and sometimes you don't. But as I said, these are like giving just AZT alone, and we saw resistance to that. And so we hope that once we get combinations, we'll get over that.
So would you, this is probably way early, but would you think of another direction to go other than broadly neutralizing antibodies for a combo or something like that? Would this be I mean, have you thought about anything like that?
I'm not sure exactly what your
well, if there was another way, would you mix it with antivirals, for instance?
Yeah.
Okay.
So there's two things. One is that I've been talking mostly here about treatment of HIV-infected individuals, but I think there's a lower bar for resistance if we talk about prevention.
Right.
So one thing would be to use these antibodies and give them just intermittently to prevent infection. And we can actually modify these antibodies now to give them a much longer half-life, such that you may only need to get an injection every 3 months or 6 months to get protection. That would be the optimistic viewpoint. And there is actually a big antibody-mediated prevention, or AMP trial, that has started, has enrolled over 1,000 individuals so far in Africa and North and South America, which is looking directly at whether a single monoclonal antibody can prevent infection. In terms of treatment, yes, we're looking at combinations of antibodies, engineering antibodies such that a single antibody can have more than one specificity, and combining them with long-acting antiretroviral therapy.
So do you think that you might be able to use these in, well, preventatively you're thinking in terms of, is there any people that it doesn't work on? Are there a group or of people that won't work with this particular, for whatever genomic?
Yeah, I mean, it's hard to say. Certainly we haven't seen any side effects or problems with these antibodies. There's always the possibility that because these antibodies have been taken out of individuals, they've been sometimes altered just a little bit to make them manufacturable, that they may not be totally like an antibody that an individual would make. So when you give these to people, their own immune system may see the antibody as foreign and make an antibody against the antibody and clear it. But we haven't seen that yet. If we do,
this is like phase 1.
Yeah. Most of what we've been doing is what I would say is phase 1 trials.
First step in humans.
Yeah, first step in humans.
So this is exciting. I think this is something that we ought to be looking forward to. It's like the beginning of, like you say, the antivirals and so forth, but this is from a preventative standpoint, which is really important. And maybe there's questions that you can't answer, like I'm thinking about, how do you roll something like this out? Is it going to be really expensive? It's an injectable?
Yeah, well, there are a couple of things. With some of the mutations we can make to give them a long half-life, we may only have to give them every 3 months and potentially every 6 months. They are injectable. They're not orally available. But depending upon the dose we have to give, we may be able to give them subcutaneously, which means that they would be able to be self-administered. Otherwise, they'd have to be given intravenously. But once again, if there are certain prevention strategies like Depo-Provera to prevent pregnancy, where the women go in every 3 months to get their Depo-Provera shot, they could at the same time get a shot of a monoclonal antibody that would hopefully protect them from infection.
Are there any other things that you want to discuss here today? Because I feel this is really a big step forward in a whole direction, new direction to be looking at. I mean, we've been talking about monoclonal antibodies for years, but as science advances, I was just talking to Paul Bernash, it evolves, and you now have this, you can't do that. Oh yeah, you can, because now we know this. So these are the pieces that put it in perspective.
I think we're very hopeful but we know that there are many steps that we still need to go through. We know that resistance to the monoclonals is going to be a problem, but we think there are ways around that. So, I don't want to sound too optimistic and that this is a done deal. In a few years, we'll have these. I can tell you in a few years, we'll have a lot of data in people to determine whether this will work or not. And I'm optimistic that it will.
Now, will you be working on this more in-house there at the NIH, or will this be in academia, or how is that going to?
So we tend to isolate the antibodies and produce them for initial first-in-human trials at the NIH. We then partner with groups like the ACTG, the HVTN, the HPTN for larger phase 2 trials. And then ultimately, if there's something that works, we need a pharmaceutical partner in order to actually license and distribute. So we collaborate all along the pathway.
And while we're at the first steps, they're very good first steps.
Yeah.
I think that's the best way because I think we want to couch it. It's like the cure. There is no cure.
Yeah.
But we are hopeful
Yes.
that there's things down the line. And this would be, in another direction, a very important step to take because we really are looking for a good way, and like you say, efficient way, because if you only have to give it you don't have to give it pills every day.
Right.
It's very good for special populations that
Yeah.
are very needful and efficient.
Yes, and it actually gets you to the standard of care for TB of directly observed
There
therapy.
you go. Yeah, exactly. So it's all can be one place to go. I think the biggest problem with HIV in certainly third world and developing world and even middle income countries is going to a clinic, just an HIV clinic.
Yeah,
It's very it's very stigmatizing. So if there's this broad healthcare aspect, prevention, treatment for certain things, prevention for others, it's all there, all one step.
Yep, absolutely.
We appreciate your being here, and it's exciting, and I look forward to hearing more from you and from those in partnership down the line.
My pleasure. Thank you.